This is the third part as we wrap up our chapter on blood, chapter 17 of our Human Anatomy and Physiology text. Just finished looking at coagulation and how it works. Now we're going to talk about factors that promote coagulation and factors that inhibit coagulation. We'll see that there is this balancing act in your blood between pro and anticoagulant factors, so that when we need to clot blood, we just tip it one way and it clots, and then we can get that job done relatively quickly as needed. So the clotting factors themselves, all those different proteins that we saw in the chart labeled with Roman numerals or some of them named, those are just proteins, plasma proteins created by the liver. And one thing about them that's interesting is that they contain a very unusual organic functional group that requires vitamin K for their synthesis. So the liver needs vitamin K to make the clotting factors. That brings us to a quick mention of a strategy for preventing coagulation. Anticoagulation is a drug called Coumadin, which is really warfarin or rat poison in a nice medical kind of packaging that blocks vitamin K activity. So then the liver cannot produce as much of the clotting factors and we can dampen down the process. Aspirin blocks thromboxane production by platelets, and so their granules will not be able to release, or they won't be able to release that thromboxanes, which activate more and more platelets in the activation of coagulation. Another thing about the clotting factors is their enzymes. Each factor cuts off a piece of a subsequent factor in the sequence, and that's like opening the cap on a glue tube. Now that next enzyme becomes active and can cut a piece off of its substrate or target clotting factor and so forth. We get this cascade of enzymatic events. Well, in your blood are plasma proteins whose sole purpose is to block enzymatic activity. Serine proteases, these are called, and antithrombin 3 and alpha antitrypsin, as spontaneous activation of clotting factors occurs, which it does occasionally, then these proteins will capture those and block those enzymatic events so that you don't have coagulation when you don't want it. So procoagulation is just the clotting factors themselves spontaneously activating anticoagulants, plasma proteins. The endothelial cells that line your blood vessels and that are the walls of the capillaries secrete inhibitors of coagulation. So they have this glistening, slick, smooth surface that the blood just glides past and no fibrin begins to accumulate on the surface of the endothelial cells. And then finally thrombin. Whenever thrombin becomes activated and produces some fibrin from fibrinogen, thrombin also activates some plasminogen to plasmin. And plasmin is the enzyme that breaks down fibrin and dissolves it. So that's another anticoagulant property of the blood. What about disorders of coagulation? What about when it's not quite working right? A thrombus is a blood clot. And typically we think of a thrombus as a blood clot inside the vascular system, possibly within the heart or within a major artery, or even in a vein. Deep vein thrombosis you may have heard of. There's a lot of television commercials about it and about drugs that are used to prevent those events. So these disorders are called thromboembolic disorders. Why? We have thrombus formation, and then if a piece of fibrin breaks off from a clot forming somewhere in the vascular system and floats along in the blood, now it's called an embolus. And then eventually that hunk of fibrin is going to find itself in a place where the branches of the blood vessels are getting smaller and smaller, and it will crash. This sticky blob of fibrin will jam up and clog some part of the vascular system, and the downstream tissues will become hypoxic, ischemic, not enough blood flow. So if a clot forms and winds up in your lungs, that's called a pulmonary embolism, and some swath of the lungs will no longer be getting blood and not oxygenating blood, so we've lost a bunch of the lung capacity. If it happens in your brain, now we're having an ischemic stroke, and it starves some part of your brain for oxygen, and that can't be good. That means that some part of your brain is not going to be doing its job, and eventually if some of the neurons are going to probably die, and you're going to have permanent debility associated with loss of function of neurons or groups of neurons. Thromboembolic disorders. What in the world would cause a blood clot to form in your vascular system? Well, three things we can name real quick right here. Roughened blood vessels. Instead of that slick, glistening surface of the endothelial cells, when you have vascular disease, atherosclerosis, then you have rough mountain range-like plaques forming inside the blood vessels, and that tends to cause coagulation to happen. If you have burns, that causes damage to the blood vessels in the area of the burn injury, and then you could easily have coagulation happening in those parts of the body. Inflammation. If you have tissue injury or infection, then you have inflammation that triggers coagulation. It's designed to prevent loss of blood when you have an injury that breaks open blood vessels. Here's an important one. Blood stasis. That means stopping flow. We already saw that the clotting factors spontaneously activate from time to time. And the reason that there isn't this runaway domino effect causing coagulation is because partly the movement of those activated clotting factors into the larger volumes of blood dilutes them out, and then those activated clotting factors will be inhibited by plasma proteins and gobbled up by macrophages in the liver. Well, if the blood stops flowing, then you have a region in which the spontaneous activation of clotting factors leads to further clotting factor activation and we have the whole cascade of events, and we have coagulation. So people with atrial fibrillation, the atria, the small chambers on the top or base of your heart where the blood enters in from the veins, sometimes those little chambers stop contracting and pushing their blood down into the ventricles. They just start to quiver. That's called fibrillation. And blood will pass through them easily enough to fill the ventricles and still have heart function. But the atria are shaped in such a way that they have little wing-like side chambers. And blood will remain static in those little side areas of the fibrillating atria and begin to clot. And then chunks of fibrin will periodically break off and go down into the ventricle and be shot out into the systemic circulation and cause strokes and other problems, or shot out into the pulmonary circulation if it's on that side and cause pulmonary embolisms. Also, patients that have weak hearts, if their heart isn't giving good circulation in the first place and they have vascular disease making it hard for blood to get through some parts of their circulation, and then they sit for a long period of time, sort of folded over, right? You fold your body up a bit, and it tends to bend shut the arteries and veins. And so when elderly people sometimes get in the car and sit in the car for a day or even 2 days at a time, heading down to Florida or whatever it may be, when they get out, they may have a significant amount of thrombus formation in their veins and arteries, and then that can be shot out and cause all kinds of problems, strokes and pulmonary embolisms. Also, airplanes. Sitting for long periods of time on airplane rides, I think they're the largest incidence of cardiovascular events in elderly is right after a plane trip. For some reason, that's a especially bad situation. Maybe it's because of a combination of sitting for a long time, tightly folded up in those very close seats, and the changes in pressure, who knows, but it's a very bad scene. So those now you understand about thromboembolic disorders. What can we do to treat? Again, typical treatment for someone with atrial fibrillation is lifelong treatment with Coumadin. It's a tricky balancing act because it's hard to, you're way upstream of the clotting events, trying to tamper with the synthesis of the clotting factors by inhibiting vitamin K activity. Well, that's not easy to manage, and so people that have to stay on Coumadin, it's a constant lifelong balancing act to keep on adjusting and correcting the Coumadin dose to account for changes in the diet. For example, there's lots of foods that change the action of Coumadin or change the action of vitamin K or the accessibility of vitamin K and so forth. So it's just a constant battle trying to have the right amount of, you have to constantly checking the coagulation times to see if things are right. If people have a heart attack and they've had a coagulation event in a coronary artery where there's disease, aspirin is usually recommended from then on a daily basis to just block thromboxane production so that platelets are slightly less likely to activate coagulation. And so we just back off that, tip that balance slightly in favor of anticoagulation then for those people. Heparin is used clinically oftentimes to really anticoagulate when there's a major event happening where coagulation is a big problem, and so it's a major anticoagulant drug that can be used. Because of the trickiness of trying to manage just the right Coumadin concentration for patients, there's a constant push to develop new anticoagulants for long-term use in such patients with atrial fibrillation. You've probably heard of some of the names of these on TV. Xarelto, Eliquis, and Plavix are relatively new drugs that are anticoagulants. Each one has run into some problems with side effects, so they've been a little bit tarnished in some ways, but the point is they're easier to manage because they directly block coagulation instead of working upstream at the point of blocking vitamin K and the synthesis of the clotting factors. And don't forget, all medical treatment involves side effects or risks. So just because some issues have been identified with these drugs doesn't mean they're not necessarily very good potential treatments for people with atrial fibrillation. All right, last thromboembolic disorder, disseminated intravascular coagulation. Disseminated means just spread out, spread all around. Intravascular, in the blood vessels, coagulation. So it just means that thousands of coagulation sites are taking place in the blood vessels throughout the body all at once. And we're talking about people that are in the intensive care unit, usually either elderly people or people with multiple problems with their body, multiple health issues, or after a severe trauma or a major surgery, they're clinging to life or struggling to hold them together, and then they develop this life-threatening DIC. So probably about 70% of people that develop DIC in the context of multiple health problems or traumas will expire from at this point. So this is kind of an end-of-life situation, very dire kind of a situation. So again, we have lots of coagulation. It causes the blood to become like sludge. It becomes very, very hard for the heart to push the blood through the circulation. Patients really not getting the kind of oxygen delivery they need right at the time when there's all kinds of problems. There's probably going to be infarcts, death of tissues in various organs, organ failure, and possibly death. An irony is that once DIC has been taking place for a while, the clotting factors are relatively quickly exhausted because it's happening throughout the circulation. Then there are no clotting factors, so we go from too much clotting over to a major bleeding disorder, and in a real problem trying to stop bleeding. There's probably been a surgery or some major traumas there's a lot of stitches and things and blood just leaking out everywhere. How about bleeding disorders? The opposite problem. Instead of coagulation, thrombus formation, we have uncontrolled bleeding. Thrombocytopenia, not enough platelets. Thrombocytes, clotting cells, that means, are called platelets, and if they're insufficient in numbers, we're going to have bleeding. So they're the activators of the intrinsic pathway of coagulation, and they're very important in the context of hemostasis. What would cause thrombocytopenia? Certain cancers result, right? Cancers in the bone marrow, we'll talk about in a bit, lead to not enough platelet production. Radiation treatment for certain cancers may result in destruction of bone marrow and loss of platelet production. Certain drugs tend to be hard on the hematopoietic pathways of making cells in the bone marrow and result in multiple problems, including thrombocytopenia. Immune hypersensitivities. I was just informed recently by a student about a really intriguing and scary situation in which excuse me, her mom has had a spontaneous episode of thrombocytopenia, life-threatening episode of thrombocytopenia, probably because of an immune hypersensitivity. The immune system was aberrantly attacking the platelets and causing them to be reduced in numbers and causing problems. So luckily that situation has been resolved, and it's a great answer to prayer. Impaired liver function can cause bleeding disorders, right? If all the clotting factors, the proteins in your blood come from the liver, and the liver is not able to produce them, then obviously you're going to have clotting problems. If you have a vitamin K deficiency in your diet, vitamin K is usually produced by intestinal bacteria or something that's supplemented in your diet. And if you have intestinal inflammation, if you're having malnutrition in general and you can't absorb these kinds of things, or they're just not being produced, you don't have the right flora in your intestine, or clotting problems. Some newborns will wind up with bleeding disorders when they've been treated with antibiotics. They can't produce enough clotting factors. Their liver isn't really fully online yet as it is, and we're sort of knocking it back on its heels. Liver disease, very common. People with long-term alcohol abuse wind up with this end game of cirrhosis. That's the late stage of liver destruction through chronic inflammation in the liver. So now we have a scarred, stiff, hard liver with very little liver function, not producing enough clotting factors. One of the myriad problems people with cirrhosis have is bleeding disorders. Hepatitis just means inflammation of the liver. Usually we're talking about a viral infection, one of 5 different viruses that can attack the liver. When there's liver inflammation, then perhaps we're going to have a reduction in all aspects of liver function, including clotting factor production. And finally, hemophilia. Hemophilia is a hereditary abnormality in which there's a missing gene so that the person's liver cannot produce one of the clotting factors. That cascade of clotting factors depends on every single one in order to continue through that domino-like effect of finally producing prothrombin to thrombin and then forming fibrin. So any one of those that are missing, there's not going to be blood clotting. So the most common genetic anomaly that leads to this is an absence of factor 8. Also factor 9 and 11 may be absent in people with a genetic disease, hemophilia. Nowadays, luckily, with the advent of molecular biology, we can produce these purified clotting factor proteins in the factory, in a factory laboratory, and make them available for infusion into patients that have hemophilia and hopefully have a relatively normal life. So some big changes just in the advent of molecular biology, that hemophilia went from being an absolute death sentence to being a chronic disease that can be managed relatively well. Okay, so that brings us to the end of our discussion of bleeding disorders. Let's quickly talk about white blood cell disorders. We talked briefly about the white blood cells and their functions. Well, if there's not enough white blood cells, it's called leukocytopenia. Just like thrombocytopenia means not enough platelets, leukocytopenia means not enough white blood cells. You can see that same suffix on here. So that's the condition we have there. It's often shortened to just say leukopenia. It's a popular term used to describe this condition. So we don't have enough white blood cells. We're going to be subject to a lot of infections in the body because of an absence of these defensive blood cells that prevent infections from taking hold. Some chemotherapy drugs are very harsh drugs for blocking cells proliferation. So sometimes when we can't find any other treatment for a cancer, we're just going to stop the dividing of pretty much all cells for a period of time to try to knock back the tumor, and it subsequently will knock back production of epithelial cells, hair follicles, hair falling out, and also the production of anywhere in your body there's a lot of cells being made, such as the red bone marrow. Leukemias are cancers of white blood cells, overproduction of a particular white blood cell or white blood cell progenitor in the red bone marrow. Could be anyone. Could be myelocytic leukemia, meaning a lineage of monocytes or any of the other white blood cells besides lymphocytes that would be classified as a myelocytic leukemia. If it was lymphocytes lineage that was involved, it would be called a lymphocytic leukemia. There's two kind of situations that give rise to the specific names of these leukemias. Some leukemias suddenly appear and the cells grow like crazy, very quick bloom of abnormal cells. That's called an acute leukemia. So we might have an acute myelolytic leukemia, AML, or an acute lymphocytic leukemia, ALL. Or some leukemias advance very, very slowly, and the abnormal cells accumulate in the blood with time very, very slowly, a chronic myelocytic or lymphocytic leukemia. Very different approaches to treating those things. Unfortunately, what happens when you have a cancer in the red bone marrow of any kind, the abnormal cell production, this overblown cell production, tends to squeeze out the production of all the other cells. So we're going to have, for example, in the case of leukemia, we're not going to have whatever other white blood cells should be produced. No, they're not. not enough red blood cells, we're going to have an anemia. Not enough platelets, we're going to have thrombocytopenia. So leukemias and other cancers can result in absence of white blood cells and susceptibility to disease, vulnerability. Well, the treatment historically has been to find drugs that will kill those abnormal cells without destroying the rest of the cells in your body. And that's a pretty great trick, right? And so we've made a lot of progress in finding drugs that will knock back those blooms of abnormal cells. But the problem is, after those treatments and the person is in quote-unquote remission, then relatively soon thereafter, the cells may come back and just grow back into this massive population of abnormal cells, and we have to try it all over again. And oftentimes, after multiple cycles, the cells may become genetically modified and more aggressive, and people usually die from massive infections during an attempt to knock back this growth of cancer cells or fight infections that happen as a result. The newest theory or strategy for tackling leukemias is the discovery that the bad cells at the heart of this problem are stem cells. Stem cells that become transformed into cancer cells, so that when they produce their offspring that then are progenitor cells that give rise to the large numbers of normal cell production, you get cancer cells, huge numbers of cancer cells. We're trying to find drugs, find ways to identify and isolate cancer stem cells and find drugs that will specifically, or strategies to specifically target them and kill them without destroying the rest of the bone marrow. Bone marrow transplants or stem cell transplants are another approach. We can, I don't know if I put anything on here, we may kill all of the patient's existing red bone marrow, essentially, and then repopulate that bone marrow by infusing them with a population of cells from a normal donor. And it's a tricky balancing act there to insert basically someone else's immune system into the body without causing it to attack the new host. And you can have graft versus host disease is called when that happens, when the new immune system begins to develop and build and grow, and then it takes turns on its own host in the body into which it's been introduced. But sometimes it's a life-saving approach. It's an improving scenario that hopefully, as we continue to improve our technology, will allow us to treat leukemias more effectively. All right, quickly, lymphomas. Lymphomas are white blood cell cancers that arise anywhere else other than the red bone marrow. So if we have white blood cells beginning to grow aberrantly in the lymphatic system, in a lymph node, for example, we may have Hodgkin's disease, solid, hard masses of cells, tumors within the lymphatic system. And they tend to stay clumped or aggregated together, and they can be removed surgically. It's a relatively treatable disease. There's a whole family of non-Hodgkin's lymphomas, they're so-called, any other type of lymphoma, in which the cells do tend to spread throughout the body. They move throughout the lymphatic system or different organs, and they can eventually move back into the bone marrow and start taking over there and producing this overwhelming growth that blocks out the production of other cells in the bone marrow and so forth. So lymphomas are just cancers that form outside the bone marrow. Infectious mononucleosis. Many of you have probably, I know you've all heard of this, and some of you may even have had infectious mononucleosis. Probably the majority of us have been exposed to Epstein-Barr virus, but most people don't even know they've ever been exposed to it, or if they do, they might have had what they thought was just a regular cold, and their immune system battled back this virus. But human herpes viruses are an unusual family of viruses that tend to exhibit latency, and people that have had infectious mononucleosis, a more aggressive form or sickness caused by this virus that can last for months at a time, can later in life develop chronic fatigue syndrome and other kinds of syndromes that are probably related to the reemergence of this virus or permanent changes in the body anyway as a result of viral infection. So there's a poorly understood syndrome that is very, very debilitating and lifelong in people that have, again, there's been an association with a previous infection with a fairly serious previous infection with Epstein-Barr virus. Okay, we wrap up our chapter by talking about blood group antigens, and I know many of you may know something about this, quite a bit about this. We've talked about it in class, but I'm just going to quickly run through an explanation of blood group antigens, blood types, and the relevance of this knowledge to giving blood transfusions safely to people. All right, so on your you have 23 different types of chromosomes, 46 chromosomes each in homologous pairs, almost identical pairs that you receive from each of your parents' gametes. So each one of those homologous chromosomes has what are called alleles, places where genes may exist or may not for producing certain membrane proteins that appear on red blood cells. So some people have in those locations, a specific location in 2 of their homologous chromosomes, alleles that produce plasma membrane proteins that we're going to call antigens, and we're going to name them antigen A, that's drawn like little triangles here, and antigen B. It just means that when those people produce red blood cells, on their surface may be found these proteins. Some people have only the allele or the gene to make antigen B, which we're depicting as little green spheres right here, little green balls. And so when they make red blood cells, that plasma protein, antigen B, appears on the blood cell surface, but not antigen A. The funny thing is, those people have an antibody, a defensive protein against antigen A in their circulation. So that if any blood cell were to appear in their body with antigen A on the surface, these antibodies would bind, and then there's systems in your blood that will lyse or pop those red blood cells and destroy them and cause problems. Some people only make antigen A, not antigen B, and therefore they have antibodies against antigen B in their circulation. And finally, people with type O blood have neither allele. So they don't make either the antigens. Their blood cells are blank in that sense, but they make antibodies against both of those two antigens. So now they have antibodies against A and B. So why do we care about this? Well, think about what blood type could someone with AB blood receive. We're talking about AB blood. They have both antigens on their surface. The only reason you couldn't give someone a blood transfusion of a particular type of blood would be that the blood would react with the antibodies in the circulation. These people have no antibodies against either antigen A or B, and so they can receive any blood type. They're called the universal recipient, and they're very lucky indeed. Hopefully they're not lucky if they have a trauma, but if they do and need a blood transfusion, then they can receive anybody's blood, just about. People with type B blood can receive any type of blood that doesn't have A antigen on its surface. So what can they receive? They can receive type B blood, right, like what they have, or they can receive type O blood that has no antigens on the surface, and then there's nothing to react with this antibody against A. People with type A blood can receive A or O. People with O blood can only receive type O blood, and that's kind of bad because O blood is kind of rare, and therefore these people are limited in what blood stock may be available to them if they're in the hospital and have a surgery or whatever, they need some blood. But on the other hand, people donating blood, people with type O blood can donate their blood to anyone. It's called a universal donor because there's no antigens on the surface. I know what you're thinking. When you're thinking about who can donate to whom, don't think about the antibodies in the donor's blood. You're only giving a tiny amount of it. It's not going to matter. We only need to consider the antibodies in the recipient's blood. So people with type O blood can donate their blood to anyone. So that's the universal donor as opposed to type AB as the universal recipient. So just remember, when you're giving blood transfusions, you can never have an antigen on the surface of the blood cell that you're giving that will react with antibodies in the patient's, the recipient's circulation. And here's just a little picture of an idealized test for blood group antigens. What's happened is a drop of blood has been placed, a couple of drops on a little plastic card, and then has been mixed with antibodies from the laboratory against the blood group antigens to see if they react. If they react, they form this clumping effect. If they don't react, nothing happens, and we know that that antigen is not present. So here, antibodies against antigen B clumped, therefore this person had antigen B on their blood cells. Antibodies against antigen A clumped, so now we know this person has type AB blood. They have both A and B antigens on their blood cells. This person only had antigen A on their blood cells, which clumped with the antibody against A. No reaction with the antibodies against antigen B. This person has type A blood, and so on. I know you probably, I hope you tried this in the blood lab last week, I think it was. The kits aren't quite as reliable in terms of producing this wonderful and discreet looking, clumping effect that we wish we'd see, but it is still fun to do. And sometimes they work relatively well, and you can learn your blood type by testing with the antibodies. Okay, that brings us to the end of our chapter on blood, and we'll meet next time to talk about chapter 17 about the heart.